Thank you, Patrick, for the very kind introduction. Very good afternoon to everybody here. Super thrilled to talk about our company, Breaking Ink, and how we are forging ahead to solve this problem around plastic pollution. If we just look around ourselves, we will realize how useful and a ubiquitous item plastic is, whether the clothing we wear, the coffee containers that we use, the plastic bottles right here, so on and so forth, the Tupperware containers that we use for food packaging, etc. So given that plastic is so useful and easy to make, it's obvious that it's just being made more and more of every single year. So just to give you some metrics here, every single year, more than 400 million tons of plastic is produced. Now, where does this all go? That is the real challenge here. Most of this, 5,000 million tons to be precise, ends up in landfills and in oceans. Now, that's not even the problem. So just by sheer legacy, we have tons of plastic already sitting in the plastics and oceans and environment. But the fact that every year more and more is being produced, and the fact that less than 10% is actually being recycled, this adds on to this monumental issue. The other half of this issue is when plastic sits in the environment, it takes tens and hundreds and thousands of years for it to go away, whether it's a plastic straw, whether it's a fishing net, or it's a diaper, it just sits there. To add to this layer even further, when the plastic comes in contact with environmental factors like the UV light and other water, rain, etc., then these plastic pieces break down into further smaller and smaller particles, which are known as microplastics and nanoplastics. Now, once they break down into such tiny particles, it becomes even harder to capture them, recognize them, and then degrade them. So that's where the status is as of today. Growing concerns about this plastic in the form of micro and nanoplastics have been emerging just in the last few years. So I'm sure most of you might have come across research studies that point to the fact that these microplastics are now entering into human bodies and also into the animals. So microplastics have been detected in almost every single organ of the human body, whether it's the lungs, placenta, breast milk, the heart. They have even known to be able to cross the blood-brain barrier, which is a huge issue. Now, what happens to all of these? Yes, there's a lot of studies going around. They have been implicated in hormonal disruption and also some forms of cancer. I'm showing you an interesting slide here where this is basically a herd of elephants entered a landfill dump yard in Sri Lanka, which is an island off of Southeast Asia. And they ended up eating plastic in the form of grocery bags and so on, and all of them died. And this raised a lot of alarm and concern in this country. So this is just one such example. And given the rising need for a solution, there is legislature push that's coming on to solve this problem. It all started when my co-founder and I were working at Harvard at the Wyss Institute, and we got interested in trying to understand how has nature figured out a solution to this problem. Now, plastic consists of these carbon-to-carbon bonds that are linked together in the form of a long chain. And the question we asked was, are there any natural microbes out there that have the inherent ability to break down these bonds? So we went on a little mission and we stumbled upon this microbe that we call X32. Now, X32 remarkably had the ability to eat through different forms of plastic. So first we started with the easiest form of plastic, which is PET. These are our PET bottles. And when we tested the growth of these against them, X32 was able to grow on these plastics. And then we tried nylon. So we basically pulled things off of the shelf like paintbrush bristles, dental floss, piece of nylon membrane, and even in these conditions, the microbe was able to grow. And then we took it a step further, and then we said, what about the more very, very tough polyolefin plastics? And there is absolutely no commercial enzyme or microbe available as of today that can break down tough polyolefins. And when we tested X32 was able to grow a little bit, but they were able to grow on these tough polyolefins as well. So immediately we realized this definitely has a huge global impact and there is a need. So we turned this into a company. We are based in Boston, and we have about 20 full-time scientists working very, very hard, to get to multiple solutions for this problem. As of today, we have decoded the genome of this microbe that we call X32, and we turned it into a highly versatile platform solution to solve this problem. So X32 is now engineered so that it can grow under various conditions, various pH, various temperatures. We can put enzymes in and out of this system, and basically it's become a highly tunable microorganism. And the idea is to be able to deploy this for several use cases. Here's a very high resolution image of a picture just to give you a glimpse of what really happens. So on the left hand side, you can see a piece of plastic. It's just a nylon film. And on the right side is the same film, but now it has microbes growing on it. And you can see in the enlarged insets that we start seeing these surface aberrations and indentations that represent that the microbes are able to chew and break the bonds of this plastic. Of course, in today's day and age, AI can't be left far behind. So in order to expedite the plastic degradation efficiency of our enzymes and microbes, we are turning towards machine learning and AI, where we take the data, we run it through our AI embeddings and large language model pipelines, and then we get some sort of candidate genes and enzymes. So these are further taken into the wet lab, we test them in the lab, and then it goes through a loop until we get to the final top hit candidates. Now, why do we want to do this? So basically, we want to get enzymes that can work across a host of different conditions. For example, those that can work at lower temperatures, those that can work on post-consumer waste, and those that can work on several different plastic types, not just one. So we are carefully monitoring the active sites of these enzymes and further iterating them so that we have a highly tunable platform here. Now, the advantage of getting these tuned is that it helps us in lowering the OpEx of all these deployment opportunities. So really, our mission at Breaking is to break down microplastics and nanoplastics in environmental remediation, which is both soil and water. So to just give you a glimpse of why this matters, so wastewater, for example, let's think of laundry and the clothing that we have. So once we do our laundry, all the microfibers that are leaching out through this ends up in the wastewater and other water bodies. And eventually, this is leaching out into the riverways and the oceans. So there's really no way to combat this and get rid of the microplastics. Once it's in the water, then it's taken up either by the fish or other marine animals, and then it makes its way back into us in some form. So this is just showing you, regions of how in Europe, how much of microplastics is entering just through wastewater bodies. So the way we want to tackle this is deploy our microbes and enzymes so that it's an additive at these wastewater treatment plants so that we can completely remove and get rid of the microplastics. Of course, eventually we want to get to land cleanup, where there's a lot of room for this problem as we know it. And then, of course, there's a ton of plastic. You might have heard of the Great Pacific Garbage Patch, and so we want to get to landfill and oceans as we have, keep chugging along and developing our platform. So with that, I would like to thank you for your attention. Happy to take any questions. Thank you very much.
Thank you very much. So the living is helping the society to be more sustainable. Can you come back on the degradation products by the microbes? I understand you take a plastic, the microbes eat it, it degrades it, it produces a couple of things that are, I believe, water, biomass, but also CO2?
Yes.
What do you do with the CO2? Because you want to get rid of it as well.
That's true. So in the grand scheme of problems, right? So the carbon in the plastic is basically getting converted to CO2, water, and biomass. So there are a number of ways we can use this. So one is we can capture that CO2 and use it elsewhere. We can, for example, turn it into another commodity like methane, and that can be used. So there are ways of upcycling, the byproducts.
And at the level of development that you are right now with breaking, are you starting to partner with some plastic manufacturers or to recycling stakeholders? Well, what are your plans there?
Yeah, so we are partnering with a lot of multinational companies, and this ranges from textile to automobile to ag tech.
Textile is a big...
Yeah, so, and because plastic is everywhere. But our mission for the moment is not really recycling it. We are more into the remediation process because it's very hard to capture the micro and nanoplastics. So like wastewater treatment, for example.
Okay. Now let's switch to the room. Are there any questions for Sukanya? Or is it crystal clear, or should I say plastic clear? Sir.
Could you speak to the self-propagation properties of this material?
Yes. So yeah, it is a microbe, and you're absolutely right. It does self-propagate. But at the genetic engineering level, we are installing safety mechanisms or safety locks, as we call it, such that when the microbe comes in contact with a certain environment, they will just die on its own. The second answer is we are starting with closed containers where environment is strictly controlled, and which is once that is established, then we'll go out to the outside and the open.
And also a last question that I had, because you mentioned the microbe and plastic, but I understand there's like whole sets of families of plastics.
Yes.
What is the efficiency of that microbe depending on the subfamilies of plastics?
Yes.
And I guess the industrial footprint that we have.
Yes. So you're absolutely right. So it starts, PET and nylon they fall on the easier bonds to break, and as we move up the plastic ladder, there are polyolefins, which are some of the tougher, harder bonds to break. So just like everyone else, we are starting with the easy ones, which is PET, then nylon. And parallelly, because our microbe has rudimentary capabilities, so we're using the genome to pull out all the enzymes that make this happen, to extend to other plastic types.
Okay. If there are no other questions from the room, let's thank warmly Sukanya for her talk.
Thank you.
Thank you very much. Thank you very much.